Hi there, my name is Adrian Monthony. I am a PhD student at Laval University in Dawood Torkamane's lab. I'll be talking today about my research studying cannabis sativa and the role of ethylene-related genes in cannabis sexual plasticity. So sexual plasticity refers to the ability of an organism to change its sexual phenotype in response to environmental or physiological cues. So what that looks like is, for example, if you have a female organism, it would be producing male sex organs or vice versa. And so in cannabis, this has been documented in response to a number of chemical treatments. And particularly, this has been well documented in female plants, which have been treated with oftentimes a chemical called STS, which inhibits ethylene signaling, and a female plant that has been treated with STS produces male flowers, as you can see here in figure 1. And so this is what a typical male flower looks like. So you can see that the flowers organs are the same, but the kind of shape of the flower resembles much more of a female plant. And so this has led us and many people to kind of believe that ethylene is acting as a feminizing agent. So in female plants, you would have high amounts of ethylene, and in induced male flowers or male plants, normally male flowers, you would have low amounts of ethylene. And so no one's really been able to quantify ethylene or look at ethylene gene expression or ethylene-related gene expression in cannabis because we don't have a pathway for this. And so one of my projects is constructing the ethylene biosynthesis and signaling pathway, as well as the Yang cycle pathway, which is a precursor pathway. And so what I'm going to talk about here is that, plus a little bit of differential expression analysis using existing data sets to try and better understand how these genes are changed based on the flower type. And so I used some existing transcriptomes from floral tissue of male, female, and induced male flowers. I took them from these two sources here, and basically what I did is I used the Arabidopsis thaliana ethylene synthesis and signaling pathways to find homologs in cannabis and construct those pathways. And so you can see here in figure 2, I was able to reconstruct the precursor cycle, the Yang cycle, the biosynthesis, and the signaling pathways. And at every step of the pathway, there is a gene or many genes that are conserved. So there's no gaps in this pathway reconstruction, which is really exciting. And I kind of want to step further and looked at where in the genome these genes that I identified in cannabis were located, and you can see that they are kind of found throughout the genome, not just on sex chromosomes, which makes sense since ethylene is involved in a lot of other processes other than just sexual plasticity. And you can't really see just because of the size of this recording in the poster, but this graph here compares the 4 categories of ethylene-related genes that I looked at and whether or not their kind of distribution on autosomes, which is the green bar, and in this stacked bar graph, and the orange shows the sex chromosomes. And so what this basically illustrates is that most of the ethylene-related genes are actually not on the sex chromosomes, it's on the autosomes. And so what that tells us is that genes involved in sexual plasticity and controlling this sexual plasticity are not simply located on the sex chromosomes. And so lastly, I looked at differential expression in those genes and was able to identify 2 motifs, which I don't really have the time to get into, but one motif of differential expression that what we called sexual plastic differential expression that showed sexual plasticity, and you can see this back and forth arrow here to the Mars and Venus symbols. And the diverging Mars and Venus symbols indicate genes that are involved in sex determination, but not sexual plasticity. And so I encourage you to follow me on Twitter because I'll be posting the bioRxiv paper for this soon. But in the meantime, I'll just end by saying that we were able to show that there are that the ethylene-related genes and pathways are widely conserved in cannabis, and that there are genes that have patterns of expression specific to sexual plasticity and some that are specific to sexual determination. Thank you.